So differential RNA processing. Here you can see after the formation of that nuclear RNA molecules, this has to be converted into functional mRNA. That means the exonic sequence should be cleaved from the intronic sequence. But in your differential RNA processing, what exactly happens? There is different kind of combination of exon together to form different kind of protein structure. I mean different kind of mRNA that will give rise to different type of protein molecules. So here you can see that a nuclear RNA which transcribes all those intronic and exonic sequences. Then by using some spliceosome, the spliceosome is nothing but the protein sequence, I mean protein, and with some RNA sequence complex combined to form this spliceosome complex. They recognize this and they can recognize the conserved sequence that found in between this intronic sequence. So whenever there is a spliceosome, it attached with the sequence complemented to that intron, it cut in this 5' and 3' region of this intronic sequence and then ligase those two ends of the exonic sequence, and that cause this formation of your functional mRNA molecules. So here in this case, you can see that different combinations of gene is being mRNA, functional mRNA is formed by using this alternate splicing, or what we call differential splicing. You better remember this alternate splicing name as it is most commonly used. Here is the alternate RNA splicing, and you can see here the consensus sequence at the 5' and 3' end of this intron. Consensus sequence and conjugate sequence are different. If I've told you conjugate sequence, sorry for that, it's your consensus sequence. That means it's very constant, means it's a kind of sequence which is constant in all those species, and it undergoes very little modification during lifetime or in evolution. But conjugate is very stiff. It won't change even in very long time period of generation by generation. Rather, it still remain as a conjugate because it plays a very important role. These sequences are. Okay, so here in this case you can see the spliceosome, which is a spliceosome are made up of two small nuclear RNA, what we call the small nuclear RNA, and a protein called splicing factor that binds the splice site or the area of adjacent bias. So here's the spliceosome, which are made of your what we call small nuclear RNA, because it has to cleave this nuclear RNA molecule, so it has much complementary sequence to identify those consensus sequences at the 5' and 3' end, and by with help of this splicing factor it can cleave and ligase those ends of this exon by removing these intronic sequences. Yes, this is the thing. As you know, the splicing factor 2, SF2, is also known as your alternate splicing factor. The choice of alternate splice site is often controlled by the which splice site can be bind a protein, and this is how it is controlled. The spliceosome forms when the proteins that accumulate at the 5' end of splice site can contact those protein bound to the 3' splice site. Once the 5' and 3' end ends are brought together, the intervening intron is excited, and the two exons are ligated together. As I told you, this is a thing. Let me show some picture of different types of alternating splicing. Here you can see it is a type of cassettes, exonic cassette, exonic type of found in procollagen. Here you can see this is a mature chondrocyte protein, where this is a precursor chondrocytes combination of this, and here is a mutually exclusive exonic. This is your limb ectoderm, this is your limb mesoderm. FGFR2 transcription factor ring form protein. So 7, 10 terminal both sides same, but the 8 and 9 is this kind of combination. This is your mutually exclusive. That means the 7 and 10 are mutually inclusive, whereas your 8 and 9 is exclusively found in those two regions. Then your alternate 5' splice. That means in the 5' region it gets cleaved in one portion, but in the other region is all these are taken out. Similarly, in the 3' alternate 3' site, the 3' site is only being taken out, but the 5' site remain as such, and in this other case both are taken out. This is a chondrin with a wild type having both the type, and the truncated variant is only taken the 3' site region. This is also it's some disease related to kind of something like that. Not so important. Okay, so here's the important thing. We have here about the exonic sequence have deals with all the protein and other epigenetic characteristics, but that's not true. If even in intonic sequence it get mutation, that means it also sometimes cause leading to some kind of physiological defects. For example, in this case, you can see your muscle hypertrophy. Here in the left is your normal time, in the right side is the muscle hypertrophy. And here what exactly happened, this gene which is responsible for the expression of a myostatin protein, the myostatin protein suppress this expression of this muscle formation. And this myostatin gene is if and in this muscle hypertrophy what happens, the myostatin gene get mutated by this intonic sequence mutation. Somewhere it must be 10. I want to find able to find. If you see, thank you very much. So forget about this. Also see, so here the region in this termination codon of this consensus sequence there is a mutation. That's why the splicing is not possible, and that causes mutation cause some. So you just said because it get inside this functional mRNA, this introntic sequence, because the spliceosome is unable to recognize this consensus sequence as it gets mutated in this guanine region, and it becomes adenine. And as we know, the AAT region is your what kind of region? This is your stop codon type of stop codon region. So when it undergoes the protein translation to form protein, it gets stopped because it is your stop codon, which is because of this mutation, and that's caused this the myostatin protein unable to synthesize, and this causes the muscle hypertrophy. Let me show you. Yes, here it is, the myostatin, the misspliced RNA. This means this nuclear RNA is misspliced because there is a mutation in this region, in this introntic sequence. The introntic sequence has consensus sequence which should be recognized by spliceosome, but as there is mutation, it is unable to recognize this mutation consensus sequence, so it takes it as an exonic sequence and leave it. And thus in this functional mRNA, when it gets undergo translated, it will form protein, but by mistake it's making a stop codon sequence. That way the synthesis protein gets stopped here, and that caused this myostatin, the negative growth regulator myostatin in this muscle cell is unable to form, and that caused the uncontrolled growth of muscle cell. This is what we call the muscle hypertrophy. And then the differential mRNA longevity, well, the longevity depends upon your polytail, the stability also depends on the poly A tail region, the sequence UTO region which we code this region, then some other protein. Okay, so here in this picture a graph is shown here. Whenever there is a prolactin, prolactin mRNA, functional mRNA is there, the casein protein is always formed. But whenever the casein protein, whenever there is let me show you the degradation of casein mRNA in this presence and absence of prolactin is shown here. So this is a percentage of initial level. So when there is no prolactin there, the mRNA which is code for this casein protein, that that mRNA is lower in amount. But whenever the hormone prolactin is secreted, or here in the experimental basis the prolactin is added, it's found that the casein mRNA remains more stable, and its longevity increases about 22.5 hours from that of your 1.1 hour. As you can see here, in the absence of prolactin, the casein mRNA decay rapidly within a half-life of 1 and 1.1 hours. But when the same experiment was done in a medium containing the prolactin, the half-life was extended to 28.5 hours. That means you can see how the longevity also regulated by different hormones, different factors like poly A tail and other protein. This is how longevity regulated. As a masking effect here, this is the O site. That means mRNA is brought together by masking the protein that binds CPP on together. You can see here the masking blocks the initiation of transcription, and by preventing this binding of E to EGF. So here in this picture you can see this masking helps to prevent, and that's how it increases this longevity of this region, or else it will undergoes degradation. So here is a cytoplasmic polyadenosine element binding protein. This causes binding to this polyadenosine site. Not so important. RNA interference in next video. Thank you very much.